people who have gone into space than have lived underwater.
It's a really important mission because nothing has been done quite like this.
Fabien Cousteau is leading a 31-day mission in the world's only underwater laboratory, Aquarius. And we're delighted to advise him on some really exciting science that's going to take place.
There are 4 specific areas where Northeastern is working. The first is looking at the health of our oceans. How can we understand what state our oceans are in? The second is, how can we use that information to live sustainably? Benefit from the ocean, but not damage it. The third is, how do we rekindle that sense of excitement in the next generation? And the fourth is celebrating the role of women in science.
I heard a statistic the other day that only 22% of the aquanauts that have lived underwater have been women, so I can't wait to spread the word and hopefully inspire young women to join marine science.
By having Liz living underwater, we can really get all these really good 24-hour measurements with the coral and study these giant sponges for a very long period of time, and you can't do that out of other platforms. And so we really get a good idea of what's going on with the human-ocean connection through this project in urban coastal sustainability.
Northeastern is dedicated to what we call use-inspired research focused on solving real-world problems. Climate change is a real-world problem. So this gives us an opportunity to engage in something that we're committed to and also to contribute to the fundamental knowledge.
Okay, so I just want to point out a couple people you saw in that video there. So you saw 2 women in there. Liz McGee is one of the women who's staying down there for 2 full weeks, or she's been there for almost 2 full weeks now. And they're coming up in just a couple days. They're at the very end of their mission now. So we may see Liz swimming around in the water at some point. But one of the women you'll be talking to, Sara Williams, was the other woman that you saw in that video. She's staying topside, but she's coming down an hour at a time here and there throughout the 2 weeks that Northeastern is spending out there. And so we'll get some of Sara's times along with another woman, Amanda, who will be down there. So now we'll hear from Isaac for some background about the science and the mission.
Hi, you all hear me fine? Okay, good. I forgot to test this before we started. All right, so everybody welcome. You all saw the video introducing Mission 31. This is a nice, good view of what the habitat looks like. It sits at 63 feet just off of Key Largo in Florida. It's the last remaining ocean habitat on Earth. There used to be a number of other habitats supported by various other countries and science programs, but this is the last one left. And as we heard, Fabien Cousteau is trying to one-up his famous grandfather, Jacques Cousteau, by adding an extra day to the 30 days that Jacques Cousteau stayed in his habitat in the Red Sea. This program is not done by one group, it's done by many. Obviously, Museum of Science is helping us out a lot by having these nice broadcasts and presentations. Northeastern University is where I'm coming from, and then all the others that are helping out with funding and other researchers. So what is Aquarius? Like I said, it's the world's only remaining underwater research habitat. What they do there is something called saturation diving. Normally when you scuba dive, you're limited to the amount of time that you can stay down based on how much nitrogen you absorb when you breathe. But if you live down there, you still breathe in that nitrogen, but it doesn't come out of your blood as you come back to the surface because you don't come back to the surface. So you can stay down there longer and you don't have to worry about it as much. There's Fabien Cousteau. He's the one that's heading all of this up. And then on the bottom there you can see those are the habitats that Jacques Cousteau stayed in when he was doing his dives in the Red Sea. So, what is it like living and working underwater? I can tell you what it's like working underwater, but I haven't lived there, unfortunately. Here is Liz McGee, who we heard about earlier, taking her first dive in the helmets that they use off of Aquarius.
Seeing Aquarius for the first time today was just magical. It was awesome to be feeling like an astronaut with the hard hat and everything else, and just gliding down to the inner space station. And just looking around, I mean, there's divers, they're getting the habitat ready. The habitat itself has become a living reef. So you've got coral and crustacean around it, and tucked away in different parts and underneath the habitat, you've got lots of life around it. You know, it's out of this world and it's underwater, and it was magical, just really magical. Of course, all these things are in preparation for the big splashdown for Mission 31. We have another week to go before splashdown, where we're going to be doing this every day for 31 days. I'm looking forward to it.
Okay. Oh, here's another video. This is, went too far. Let's go back. Come on, button. There we go. Why don't you do it? There we go. So one thing you have to learn when you're scuba diving is how to get your mask clear again if it gets kicked off by somebody else. So you have to learn how to flood it and then empty it back out again. And this is Liz learning how to use the helmets for Aquarius. So what you'll see when she turns that knob on her helmet, you'll see water start to flood in. Or actually, it's flooding in now, and then she'll turn the knob to push it back out.
Oh, wait a minute. This is also the first time most scuba divers are able to physically talk while underwater. Normally with their mask and everything is they're breathing and they can't talk. They communicate with hand gestures. But with these particular masks, some of the helmets that they're using, they can actually talk and communicate with, I think it's a wireless system.
They're wired in. They've got a big
okay, so they are wired in.
Back.
And they can actually talk. And she said the first time she started talking under there, they couldn't get her to shut up because she had never been able to talk and describe what she was seeing while she was diving.
Yep. This is a tour of a previous event down at Aquarius involving one of the current members of the team, Mark Patterson.
Today our boat headed out to the Aquarius Reef Base, where the aquanauts will live, work, eat, and sleep on the ocean floor for the next 7 days. I'm a technology and oceans reporter, and when I heard Aquarius was having its last scheduled mission, I had to check it out for myself. My dive buddies were Mark Ostrich from One World, One Ocean, and Fabien Cousteau, the grandson of Jacques Cousteau. Fabien Cousteau is visiting Aquarius because his grandfather created the first underwater habitats 50 years ago. We were the first to visit the aquanauts and get a tour of the habitat as they moved into their new home.
What is all this stuff?
It's moving into 20,000 millimeters under the sea, one of the coolest pieces of real estate on the planet. We just got here a few minutes ago and now we're moving in. You can see there's lots of action in the bunk room.
A lot going on. There's a lot of pots coming down right now, all the electronics, and I'm just eating a cracker. There's actually wireless internet access, so we're underwater and there's wireless internet access. You can surf the web.
We also got a $1 million view out of this prime piece of real estate. We eat our meals here. This is a wonderful place to take in the predator show at night. But most of the time we're actually outside. That's the whole purpose of this underwater space station, is not to be in it, but to be out on the reef doing our experiments and studying this wonderful coral reef.
This is my third experience here. I hope not my last. It's like a dream come true. It's what little kids dream about. And here we are, we can be little kids realizing our dreams.
It was a really unique experience interviewing Sylvia Earle in Aquarius. She's lived underwater 9 times before this, and I think that makes her the most experienced of any aquanaut around.
The treatment for the bends.
These are all valves that control the air and the habitat. These are all switches, almost like a breaker in your house. You do not want to hit that red button right there. Don't want to hit that button. Right here, you can see us where that's actually the live webcam going out to the internet. Over here, we have our galley and our sink. We got hot water, microwave. Somebody was making mac and cheese there. So up here we have a lot of food. Yummy M&Ms. That's not a plug for M&Ms, but they are very, very good.
We've learned, how to take showers and keep the habitat nice and dry, where to hang our towels, aquanaut etiquette.
These doors all have O-ring seals. These are all sealing surfaces. We actually close these doors and pressurize the inside cabin here to a different pressure. This is what we'll decompress at the end of the mission. We'll bring the inside pressure all the way up to sea level with the doors shut, and then we'll do a very short dive to the surface.
Brian, we're going to have to wrap it up. Only 1 minute.
The aquanauts can be down there for 7 days straight because they're saturating, but since we're just scuba divers, we've only got an hour in the lab. And for that hour you have, it goes by so quick. Before you know it, it's just time to leave. Sylvia and the other aquanauts were gracious hosts, and I look forward to visiting them again.
All right, so what's the purpose of this? Well, one is to raise the attention of ocean conservation, and that's what Fabien Cousteau is all about. He's about saving the oceans, which is something we all should be worried about. But on the other hand, we also want to do some science while we're there. The big advantage to Aquarius is you can do about 60 days worth of science in a couple of days. Because normally you have to make repeated dives down and then back up and down and back up, and you're limited. Maximum the surface divers can go down 3 times for an hour each day, whereas the aquanauts, they can stay out for up to 8 hours every day. They're just umbilicaled into the Aquarius with their helmets, so they can do whatever they want. So the big themes for this trip are corals and environmental stress, environmental contaminant exposure, sponge feeding activity and the energy budget, health of reef zooplankton, which you'll hear more about today with one of our divers, and sponge DNA for the ocean genome legacy. The other person you're going to be talking to today will be covering the corals and environmental stress as well. So, what about corals and stress? Corals are very important organisms. They're what make up coral reefs. They make a lot of habitat for smaller organisms to live in. Here's a couple examples. This is a brain coral, and then this is called a cup coral. You can see they're very different, but they're the same basic organism. They're very small. They kind of look like an upside-down jellyfish or squid or something, little tentacles. They have little plankton that live inside their cells, and they're called zooxanthellae, and that's what gives them their energy. They photosynthesize the light, and they provide energy to the corals. So you'll hear about this as well as the zooxanthellae a little later. But they're very sensitive. Any sort of stress can cause problems. So what Sarah Williams and Mark Patterson are doing is they're taking this very sensitive, very expensive piece of equipment down, and they're going to be measuring what's going on inside the coral. Well, a coral's very small. It's about that big most of the time. And so what they're going to do is they're going to insert this little probe inside the mouth of a coral. Well, that probe has to be very small, on the order of about 25 microns. That's about half the width of a human hair. So this is a piece of glass that they're going to very slowly insert inside the coral, and it's just going to sit there and breathe with that thing in its mouth. And so then they can measure what's going on. And when you know what's going on inside the organism, and you can also measure what's going on outside, you can make various predictions on how they're going to change to different conditions. No, I think you were blocking it. Environmental contamination at the reef. Another big problem is we get a lot of environmental contamination coming in from coastlines. So, for example, they're down in Florida. Well, there's a lot of large cities along the Florida coast. You get a lot of contaminants coming out of there. You also get a lot of sediment, things like that. So, one thing that's going to be going on is they're using these little collection devices, and it's pretty simple. All it really is some pieces of plastic attached to a float, and those plastics actually absorb the toxins and the pollution in the water the same way that the organisms do. So what they can do is they can leave them out there, and then the aquanauts can go out and they can take a little piece of plastic every day, and every so often they take it back and they wrap it in foil and they send it to the surface. And then those plastics will be sent to a lab, and they'll analyze what sort of chemicals and things are inside them. This is a lot easier done with the aquanauts because they can go out whenever they want and get it. And so here we can see what they look like. We've just got a little bulb floating up there in the top, and then the plastics are attached to that rope. Another thing, sponge bioenergetics. So how does energy flow through the sponges? What they're going to be doing is they're going to be inserting a device into the middle of these barrel sponges, and these can be up to thousands of years old. Think of like a redwood tree, and some of them get very big. You know, you can actually get inside them. Some of them are like this big. So they have a device.
Sorry, I'm going to interrupt for just 1 second. These are who we're going to be talking to later. They just are swimming down. If you guys can see anyone, I'll put it up on the big screen later, but that's what we're checking on with this. So these are the two girls.
No, actually, that's Mark and it's Mark Patterson and Francis. No.
Oh, we're not talking to them.
Nick Kolvar.
Okay.
But they're also in those labs. So
yeah, they're part of that
lab. They're the
lab.
other support crew.
Okay, so maybe we'll see them swimming around more later. But that's just why I keep looking at other screens, because I'm waiting for those. Oh, I don't know if they can see. Oh, there they can. Okay. Okay.
So these folks they're part of the surface crew, so they only get to come down for a few hours total every day. Okay, so back to the barrel sponges. You can see what you see in this picture right here is this is someone measuring how the water flows through these sponges. These sponges will filter all the water on the reef every day or two. Like, think of the entire ocean around a reef, and they just suck the water in and push it back out. Well, when they do that, a lot of particulates come out of them, and they filter other stuff out. So they're very important to the reef. And this is another video.
Is this a video?
I believe.
Try hitting it one more time.
Coral reefs are the rainforests of the sea. This ecosystem may be our best hope for the cure for cancer, heart disease, and a host of other human ailments. We may find these drugs on the reef.
Yeah, they're getting ready.
So today what we're doing is we're making some measurements from the day boat using scuba diving. We're taking a water quality instrument called an XO2, and this is a really cool device that's got all manner of probes that can measure dissolved oxygen and pH and the temperature and the salinity of the water. And one thing we're going to try today for the first time is using it as a probe to measure the metabolism of some barrel sponges on the reef. Sponges are the oldest multicellular life on the planet. All the water on the coral reef passes through the body of a sponge every 24 to 48 hours.
The sponge communities are really important on the reef. They're a really valuable filter. As anybody who has a home pool knows, your pool will go green if you don't have a good filter cleaning the water in it. Sponges play a really important role in that. Those hard bottoms of the spurs are really where the sponges and the corals want to take hold. Sponges really like to have a nice hard substrate to anchor onto. So we're going to be looking at some of those areas and try to see if we can get a sense of what their preferred substrate environments are. Maybe we can find new medicines out there, new ways to treat diseases, even new engineering systems for filtering or help develop reef systems in other places. So it really makes it exciting.
It'd be a tragedy if we lost the cure for cancer because we lost the components of the ecosystem. So that's why it's important to understand the functioning of the entire ecosystem and protect it, because our well-being is at stake as a species.
Okay, so the next theme that we're interested in, zooplankton health. Now this is one of the things you're going to hear about from one of our divers in a few minutes here. How do zooplankton change with the effects of climate change, such as global warming or ocean acidification?
Right place at the right
So
time.
zooplankton are the small little creatures that you find floating around in the water and swimming around. You can see a video here. These are all zooplankton at night gathering on the window of Aquarius, and they're an important food source for a lot of the other animals on the reef. But they also work with the corals to survive. So knowing how they're being affected is very important, because they're one of the main links in the food chain of the coral reef. If they're having problems, everything else is going to be having problems.
Our oceans are the next frontier in scientific discovery.
So this is a little video about the ocean genome legacy. And what they're
that can
getting
provide
out of this
genetic data to help us better understand
is
our environment.
DNA samples from a lot of the organisms
Researchers here
that they will store and possibly use later on. As you heard Mark say, there could be the cure for cancer on a reef, and we don't want to lose that if we lose our reefs.
Urban coastal sustainability. Our marine science faculty and students are pursuing knowledge from the depths of the ocean to the coasts of continents, and in our state-of-the-art labs. Part of their journey is now rooted in an amazing partnership between Northeastern and Ocean Genome Legacy, a public biorepository of DNA samples from ocean life. A physical database of the world's rarest, strangest, and most remarkable ocean creatures that's now right here at Northeastern in our Marine Science Center. This biorepository provides an unprecedented wealth of knowledge for our research faculty, students, and scientists around the globe. An infinite world of discovery is waiting below the ocean's surface, and thanks to OGL, that world is now accessible in our labs.
So here's a quick picture of our current aquanauts, some of them. We've got Liz McGee from Northeastern there, and Grace from MIT, and then Fabian in the back, and then I don't remember what his name is. He's one of Fabian's cameramen, I think.
Ryan. I only know Ryan.
But some things to point out that you saw during those videos is, so the aquanauts down there, they have multiple options for going out in the water. They can use their helmets, which are actually tethered to Aquarius, meaning they have unlimited air, and they've got their mics, and so they can walk around, and they can walk around up to 400 feet away from the Aquarius habitat, whereas everybody else has to use scuba gear. Now you can see this is your air source, so obviously you've only got so much. And this is what you use to breathe. So talking is not real, doesn't go over so well. So there's a couple big differences between the surface crews, and you'll hear from some of the surface divers in just a couple minutes. They're ready for us, and what the aquanauts get to do. A quick note about this. This is a plankton net. This is what Amanda, who you'll be hearing from in a moment, is using to collect her samples. So the nice thing about marine science is you have a healthy mix of very high-tech equipment and very low-tech equipment. So without further me talking, we'll get the ladies up on the screen here, and we can start talking to them.
So I thought I'd point out a couple things. So what you see up there now is through Florida International University. That's who runs Aquarius, and they have multiple live cameras down there. And so this you can watch anytime, no sound. It's creepy but really cool to do. And so what you're seeing there, the two women seated on the right, that's who we'll be talking to. That's Grace Young walking right behind them now, and a couple of their other support crew and researchers are there. And we will put them up on the live chat in just a second. Oh, Grace is joining them too. So now I will put our actual chat up. Sorry, yes, the upper right corner. Sorry. Silly. Ah, there. Geez, I was looking at the picture bar.
Okay, they should be up now.
They sit there muting them. Can you want to start broadcast on the button? Push that button. Okay, Aquarius. Can you guys hear us? We're not hearing you. I think you're muted. You are muted. You're still muted. You are muted on your end.
Okay, can you hear us now, Susan?
Yes, we can hear you now.
Okay, can you hear me?
Sorry, the cameras don't hear, but you're up there.
Hi, guys. Hi. No, that's Fabian Cousteau. All right.
So do you guys want to introduce yourself? We've mentioned who all of you are, but if you want to just do a quick introduction of who is who there.
So, hello, Boston Museum of Science. My name is Sara Williams, and I'm a research technician at Northeastern University.
I'm Amanda Dwyer, and I'm a PhD student at Northeastern University, and we both work in Dr. Mark Patterson's lab. And I'm Grace Young. I just graduated from MIT, and I'm an aquanaut right now on Mission 31.
We are very excited to be talking to you from 20,000 millimeters under the sea at the world's only undersea laboratory, Aquarius, as part of Fabian Cousteau's Mission 31.
That's great.
Great. Do you guys want to wave? They can see. Can you see the audience? Okay, kind of.
Hi, everybody.
Yeah, we're all from the Boston area, so we've been there. We've been right where you are.
Yep. Yep. So for the past 2 weeks, Amanda and I have been a part of the Northeastern surface team working on Mission 31 science. As a research tech, I get to work on a lot of the different projects, but my main project is focused on how corals respond to environmental stress. So I've been diving down every day to the habitat with a special underwater system to measure oxygen dynamics in corals. And corals, where'd the cord go? Oh. So corals are facing a lot of hazards in the oceans today. There's global warming, or ocean acidification, and rising sea level temperatures are causing corals to get really stressed out. And we are measuring how environmental stress affects the corals. So if you guys don't know, a coral reef is formed by many colonies of corals. And each coral, a coral is actually an animal, not a plant or a rock. And each coral has many polyps, which are identical units, part of the coral. And we are looking at how these polyps are connected and how they react to environmental stresses together. They can work together to respond to stress. So my lab in particular is interested in the gastrovascular system of a coral. And the gastrovascular system is how the coral is connected by different tubes and canals. So there's two types of coral gastrovascular systems. There's an imperforate coral and a perforate coral. Imperforate corals are only connected to each other when each polyp is expanded, and only to their nearest neighbors. Perforate corals, however, have canals that connect each polyp to one another throughout the whole skeleton of a coral, so they can better share resources when they're stressed out. So what we've been doing while we're down here at Mission 31 is working with this really cool Unisense underwater meter system and associated oxygen microsensors. And these microsensors are really thin glass probes that are thinner than a human's hair. So they're very, very fragile. And what we do is we actually put these glass microsensors inside of a coral's mouth and measure what's going on inside of that coral gastrovascular system. So every day I've been diving down.
What does it mean for a coral to have a mouth?
A mouth?
Yeah.
So corals have mouths just like us. Yeah. Each polyp has its own mouth, and that's ringed by tentacles, and they can actually grab food and bring it down into their mouth. But they also have symbiotic algae that they can photosynthesize with that gives them extra energy. So, yeah, which is really cool. And the mouths are really, really tiny, and the probes are really, really tiny. So I have to use this special magnifying headwear underwater to be able to see what I'm doing.
It's really cool.
It does look really cool. I'm like especially nerdy. Yeah. So every day we go down, I dive with this special headgear, and I put the probe into this little micro manipulator, which is this fancy tool that allows me to move the probe around with very, very fine adjustments. And then I get it inside of the coral, and we leave it there for 24 hours or so to get some really cool data. No one's ever done this research before, so it's really awesome to get a really good 24/7-hour period of what's going on inside of the coral. And by looking at these oxygen dynamics inside coral, we can better understand how they're responding to environmental stressors. So one of these environmental stressors that you may have heard about is coral bleaching, which is an event where corals actually expel their symbiotic algae that live inside their skin when the ocean waters get too hot or it's too bright for them. And so when they do this, they often have to rely on catching external prey food sources like zooplankton. And I'll turn it over to Amanda now to talk about her research on zooplankton.
Yes. Oh, perfect. So that's actually one of the
Where did that come from?
Right? Yeah, we were hoping to show you guys one of these through the window, and we didn't. So it's great that you have one
Brian
there.
Helmuth through the window.
Oh, yeah, here's some more of our topside team that you can see.
And Francis Choi.
And Brian Helmuth, yep. But I use those traps to help see what zooplankton are coming up at night off the reef. So like Sarah was saying, with coral bleaching, there are some studies that show that corals can recover from bleaching events by switching their diet more heavily on zooplankton. But unfortunately, there isn't a lot of research now on zooplankton and where they are on the reef and if that's going to be a sustainable solution for corals to actually recover. So during this mission, while Grace and Liz have been down here, we've been getting different samples during the day and at night of the zooplankton. So these traps are set up by the aquanauts every night. And what we do know about zooplankton is that during the day, they stay at the bottom of the ocean to protect themselves from getting eaten by fish. And then they swim up to the top of the reef during the night when it's darker at the top, and that's when they're able to feed on phytoplankton or algae, which is at the top of the ocean. So they swim up into those jars, and then in the morning the aquanauts swim and collect the jars, and then on our surface team, when we dive down, then we collect the jars and process them on the boat during the day.
Oh, didn't you bring some zooplankton to
And
show
so
everybody?
this is what we do. Put it up closer to the camera. So this is some of the plankton that we collected today, actually from one of our surface tows, which we do. So just from the boat, we bring a plankton net and see what plankton is at the top, and we also swim the plankton net straight down from the surface and see a representation of the whole water column in the ocean. And then also during the day, the aquanauts swim a tow during the day through 3 different heights for 50 meters. So right over the bottom, and then 1 meter higher, and then at 2 meters over the bottom. So that gives us a good idea of what zooplankton are on the reef during this 2-week period, which is some pretty exciting data to have.
Right.
For lots of things, as well as just what can recover from bleaching. And there's a really, I don't know if you guys can see the eagle ray.
Yeah,
Sitting
there's
by
an
the
eagle
window.
right outside the window right now.
Oh, there's two
way
of
out.
them.
There's
Yeah,
two.
Do you guys want to take a couple questions?
Yeah.
Sure.
Okay.
Yeah, I'll take questions.
So what's your name? What's his name? Okay, we have Raphael. How old are you, Raphael? He's 4.5. And he's
Hi, Raphael.
Hi, Raphael.
very excited. What's your question?
How do you get down to the space station?
How do you get down to the space station?
So Amanda and I actually scuba dive down to Aquarius, which is the world's only inner space station. So we have a big tank on our back, which is attached to a VCD, which it's kind of like a life jacket. So our life jacket keeps our tank on, and then we swim down.
Yep, I got here the same way. We swam down with scuba gear. That's scuba gear. You guys are awesome with the props.
Thank you,
They
Ivan.
just show up in the screen.
Apparently
Okay.
that's the one from the 3 C's group. They were wondering where it was, and then it turned out we have it here.
Awesome. Yeah, that's the gear we use to get down here.
So does anyone have any other questions about them down there? Okay. What's your name?
Justin.
This is Justin.
Hi, Justin.
Hi, Justin.
Yeah. How deep is the sea going?
How deep are you? How deep is it?
See.
The sea floor where we're at is about 62 feet deep, and the habitat itself, though, is raised a bit off the sea floor. So right now we're at 46.1 feet, and there's a monitor on the wall that shows our depth. The depth changes slightly each day based on the waves and the tide above us. But about 46 feet.
there any other questions out
That-
here?
that's
Oh,
like a
yep,
fourth-
yeah.
I'm Charles, and I have a question about what location are you underwater, like near what coast or something like that?
Oh, yeah, where are you guys? We haven't said where, off Florida there?
Yeah, so we're a couple miles off of Key Largo, Florida. Yeah, off of Key Largo.
Do you guys ever swim further away to a deeper place? Like, do you ever go down any deeper than where you are? Like from Aquarius?
From our dives for the surface team, we've been around 63 feet is our maximum depth that we've been diving around the habitat. Grace, have you been out any deeper on any of your excursions?
Yeah, most of our work we've been doing close to the habitat, so we haven't gone too much deeper than like 60, 70 feet. But you can go up to a little over 100 feet around here on the dives we're doing.
Okay. Does anyone have any other questions? Yep.
What specific species of sea creatures are you studying?
Right. So right now, I'm looking at coral, which is closely related to jellyfish, actually. They're cnidarians. And they're kind of, if you've looked at pictures of reefs, there's those rock plant-like things, but they're actually animals. And we're specifically looking at Montastria cavernosa, which is called the massive star coral. And Amanda, you're looking at...
Yeah, so I'm looking at the plankton again. So these are basically, you can kind of see them, but basically microscopic animals that live in the ocean. So that's what I'm studying. And I'm looking at, I have this high-speed video camera, so I'm looking at creatures that move really, really fast, and then this special camera allows us to see their super quick motions in slow motion. I'm looking at, for example, fish fins that are moving really fast. We're trying to catch the grouper. It's a giant 300-pound fish as it's feeding.
It's bigger than Amanda here.
Yeah, it's bigger than all of us.
It's bigger than all of us.
Some of us are more than 300 pounds. They trying to film that motion.
And what are the eagle rays out? I heard they were telling us the other day there's eagle rays that
There's
have been
7 of them
circling
that are
outside of them. Have you seen them from the surface team? Have you guys seen them?
Yeah, so they're very curious. They keep coming after us.
Coming really close.
Oh, it is.
Okay, let's bring them right here. Oh, they just went over that camera shot.
Okay. Yep. Yeah.
We've seen them for 28 days straight, which is extraordinarily unusual considering they're pelagics.
Yeah, they've been circling the habitat for pretty much all of Mission 31. So they've been very curious, and they'll just come swim or circles around us all day while we're doing our dives and our science. Yeah.
So are you guys going to get to do any science down there today, or are you down there just to talk to us?
Well, today we couldn't blow all of our bottom time on doing any other science dives, so Amanda and I are just down here to talk to you guys. So it's a very special event, and we're excited to be down here.
Yay!
But we've been doing science all day, every day, pretty much for the past 2 weeks, and we do 2 to 3 dives a day, where we're down, anywhere from 30 to 50 minutes on each dive. Yeah.
So what's each of your favorite things to do down there? So Grace, you've been living. So what's your favorite? And then for the topside divers, what's the best part for you?
I really enjoy spending so much time in the water because we're saturated. We can spend hours, over 6 hours a day in the water. And I love being able to observe how the ecosystems around the reef change in the period of an hour or a few hours. And I enjoy this because I really like the research we're doing and the experience of the adventure of it all living underwater. So every moment is a big thrill for me.
Yeah, my project, like I said, a lot of it's done by the aquanauts, so I get the opportunity of helping other people. So it's really cool to see all the different research that our topside team is doing, the microsensors and getting it set up, and then Allie's sponge work and using the cameras and videotaping what's going on.
Yeah. I think my favorite part so far is actually working with my very fragile glass probes and getting it inside of the coral's mouth. It's really hard to do, especially because we've been having some very fast currents go by. And so I'm having to stand there, or I guess swim there, very, very slow and hover right over my coral, my very fragile, very expensive glass microsensors, which is terrifying. But the last couple dives, I've actually been able to get the probe tip, which is actually thinner than your hair, inside of the coral's mouth. And that's been very exciting and very different than the lab work that I've been doing.
It's fun to watch you. He's like a surgeon.
underwater.
I am. I'm like an underwater brain surgeon.
Yeah. I think they're coming up right now.
Oh.
You can see it.
Okay.
Yeah, so in a couple minutes, we're actually going to have the aquanauts bring up my underwater Unisense microsensor system.
Yep, here's Liz.
Liz Bentley McGee, our diving safety officer at Northeastern's Marine Science Center, one of the aquanauts scientists. Oh, and I see my underwater meter system coming up, almost here. So here it is. You can actually see. It's like a little box like that, about that big. And right over there is one of the very broken probes, actually. I think one of the Goliath groupers knocked it over the other day. I'm not very happy with them. And those glass tubes right there, those are the microsensors. Yeah, and then there's the underwater meter system. Yeah, where was? Hello. Yeah.
Well, we're gonna
Thank
actually,
you,
we're
Liz.
gonna wait. Hold on, Liz. If
Liz,
we were gonna
Liz,
look
Liz.
through Liz's camera for 1 second.
Hey,
Hey,
Ryan.
Liz.
Ryan, can you ask Liz?
Hold on 1 second. We can't actually yell through the
Liz,
glass.
bring
It's
that
really
back.
thick. Oh, we actually have one of Amanda's plankton traps.
Yeah. So you guys got to see one up close and personal at the museum, but here's one of the ones that's underwater.
Okay. So they can't see us so we're. Oh, actually, I guess you can see us. We're looking at through Liz's camera right now. So we're seeing you. You're waving at us through the glass.
Oh yeah. Look through the glass. I'm sure you can see it. Can you see through the glass? Yeah, so here is the underwater meter system again with the very
So it's
fragile
the
glass
exact same thing
probe.
we've got here.
Yeah. Susan, can you guys see us?
Yeah, we're looking through Liz's camera.
Ah, well then we'll wave.
So we're in the water looking in at you right now.
That's funny. Okay. All right, cool. Well, let's see if we can get them to switch it back over to our camera so you guys can see it through the viewport.
Okay, switch it back.
You see our bowl of M&Ms?
Yeah.
M&Ms! We just got real excited. It's like taunting her.
Yeah, but where is it? No, that is it. Is that it? That's not it. This is it. You got it? Okay, we're getting your picture back up, but you can still hear us.
Okay.
Okay.
Okay, well, you can keep talking to us because we can still hear you. We're just not looking at you. We were looking at the FIU website. So.
Oh. All right. So Amanda, what is?
Done with Liz? I'm done with Liz.
Yeah. Yeah. Can we switch it back to our video?
Yes, you are.
Yeah.
We've got 3 computers going, guys. Give a
You guys
They have one there. They have one. We have one there. It's okay. Thank you. No.
Take me there.
Oh, we can hear her talking for a long time. So it's been really funny this whole time with Liz and Grace on their helmets, and they'll be trying to talk to us, but we can't hear them through their comm system. They can talk to each other, but all we hear is this really high-pitched blur, like under the water. And so it hasn't been working, and underwater communication is really tough when you're just scuba diving. We have slates that we write on, but sometimes I'll hear Dr. Patterson just kind of going off through his regulator, and I can almost tell what he's saying.
Yeah, he tries to talk to us all the time.
He tries to talk all the time. Yeah.
I'm not close to.
Is your voice set higher under that? Because we can't really tell, but supposedly your voice gets kind of higher in there?
Yeah.
Yeah.
So we're breathing compressed air, so there's odd little things like we can't whistle.
What happens if you...
I can whistle. You can whistle a little bit.
Whistled.
I can do it.
Sarah can whistle.
I can whistle.
Never mind.
All right, sorry to prove that very wrong, guys.
Normal people cannot whistle.
Are there any other questions from our audience? Here, we've got one back here. Yeah?
For those living under the water for 31 days, how long will it take you to decompress?
18 hours. Yeah, actually, the decompression time is the same whether or not you've been living underwater for a day or a week or 31 days. It takes us 18 hours. During that time, our bodies become acclimated slowly from living at 2.5 atmospheres to 1 atmosphere of pressure.
Okay, we have another one here.
Hi, what do we know about the effects of human activity on the ocean ecosystem at greater depths? And could you foresee a project like this one, but that would be stationed at a much deeper part of the ocean floor?
Well, I know things get a lot harder to study as we go deeper because of the pressure and the extreme environments. And I know not much has been done, and I'm sure we don't know very much about the deep ocean right now. More of outer space has been explored than inner space. So
yeah, the deep ocean is extra challenging because the deeper you go, the darker it gets, the harder it is to reach from the surface. And right now, our air, our electricity, our internet are supplied from a buoy on the surface. And those cables, they'd have to go even deeper to get to where we would be on this seafloor. We also are dependent on surface divers that bring us food, and they help repair the habitat. And because we're 60 feet deep, they only have a bottom time of about 45 minutes. If we were even deeper, their bottom time would be like 20 minutes. So that's not even enough really to do some maintenance work. There's a lot of extra challenges. But people have lived in habitats that have been deeper. I know there was one at 80 meters. I forget which one that was.
Okay.
And there's been some deeper ones. Like commercial divers do deep work in the really deep sea in diving bells and things. So it's possible. It's not comfortable, and we definitely have a lot to do to improve underwater living.
But it would be really cool to actually have a habitat deeper in the ocean as technology
Definitely.
progresses.
Definitely.
Yeah.
Yeah, the animals living down there must be so crazy.
Yeah.
It's like nothing we've ever seen.
Okay, we've got another question over here. What's your name?
VJ. How do you get to the underwater place? Like, do you ride a boat to get actually to get to the place?
Yeah.
Yeah. So do you take a boat out?
Huh?
You take a boat out from the. You don't have to swim all the way from shore.
Yeah, no, we don't swim all the way. If we swim all the way, we'd be too tired to dive. Yeah, so we take a boat out from Aquarius Reef Base every day, and it's about a 45-minute boat ride, and it's really nice, but Amanda gets pretty seasick.
Yeah.
So she's been fighting that, but she's been a trooper getting her zooplankton samples taken care of out on the boat every day.
Yeah, so it's good that we have a boat too, because besides being a tiring swim, we do about 3 dives a day, and in between each dive you have to have about an hour of surface interval between your dives. So then we're on the boat during that time, and that's when I process my samples.
Okay, are there any other, we got a question in the back here.
Amanda, this is Emma and Simon, and we just want to say hi to you.
Hi Emma, hi Simon. Oh, fun.
Are there any other questions? Yep, we got one here.
How do you get the air supply in the actual Aquarius?
Yeah, is it sent down or is it made down there?
It's sent down. We have air compressors on the surface that will send down air. And there's also storage tanks outside that are big cylinders that store extra air. So we have our primary air supply that's in the habitat, and we have a secondary air supply just in case something goes wrong with our primary air supply. We'll be fine.
So are you mostly doing a lot of collection of data now, and then you'll go back and I guess do months and years worth of processing now and analyzing everything you've gotten?
Yeah, so Amanda started looking at some of her samples.
Yeah, so we brought shipped a couple of microscopes down. So I've done just looking at a couple samples and taking pictures of some cool things. Another thing that I'm looking at that's pretty cool is we have a dye that will stain the samples so we can tell what was alive and what was dead when we collected them before we put them into preservation. So it is known that there are a lot of so-called zombie copepods, so there can be lots of carapaces or dead copepods going through the ocean. And so without the stain, when I'm going through my samples, I wouldn't know which ones were really alive or dead. So that adds a different kind of time constraint. So I'll be hoping to get through all of my samples by the end of August.
Yeah, I guess working with live animals like that, you need to work faster.
And all of my data from the underwater meter system, I just plug in a cable and download it straight to the computer. And it's a little bit tricky. The time and date stamp has gotten a little off, so I have to go back through my notes and really rigorously figure out what's going on. But it's cool because we can see kind of the respiration and photosynthesis cycle that the corals are having with that oxygen dynamics inside of their coral stomachs really well, just looking at the data, the raw data.
There any? Oh, we do have a question here.
How does one decompress?
That's a good question. What do you do to decompress?
I'm going to take that one. So the air inside the habitat goes from being compressed at 2.5 times to the pressure of air on the surface. Slowly it comes to 1 atmosphere of pressure. So the air becomes less dense, less pressurized. And while the air is changing the pressure, we are also breathing special mixtures of gases to make sure our bodies are acclimating okay. I think we breathe extra oxygen for a little bit to help our bodies release some of that nitrogen that's absorbed in our system.
How about the surface team? Do you have to do anything special or just slowly go back to the surface and wait?
Yeah, so you swim up very, very slowly. You don't want to go up more than like a foot a second, basically, when you're swimming up from a dive. And then at about 15 feet, we stop and hang on to a line for about 3 minutes to let all that extra nitrogen and stuff come out of our bodies. And then we wait on the boat for another hour or so before we go down for another dive. If we went back right after, then our allowable bottom time would be much shorter.
Okay. Oh, question.
Can you feel the adjustment when you are acclimating back to surface air pressure? Do you feel anything like fizziness or I don't know if you burp more?
Okay, that's still enough. So my ears are pretty sensitive as well. So when I'm going back up, sometimes you can kind of feel
the pressure change.
Yeah.
Like when you can feel the change in depth, but you don't really feel the gases that are kind of going in and out of your body as you're scuba diving.
Yeah, if you're doing it right, you won't feel. If you're doing it wrong, there's actually bubbles that'll come that are compressed in your blood when you're deep, and then when you rise too fast, I heard you can like feel and hear the bubbles in your joints. But that's if you're doing it really wrong.
That's when it's called decompression sickness or the bends, if you guys have heard of that.
Yeah. So fortunately, none of us have felt that way. Yes.
Okay. Are there any other questions out here? I don't know. So anything else you guys want to tell us about what you've... are you going right back to the surface now after this?
I think we've got about 10 minutes or so to call some people or get a tour of the habitat, which will be really cool.
Is this your first time on the habitat during all your short dives?
Yeah, this is our first time inside.
Yeah, this is my first time ever inside.
And I came in and visited Sylvia Earle and Mark Patterson back in Mission Aquarius in summer of 2012. So it's really neat to be back.
Has it changed very much?
Not much. There's still a lot of buttons on the walls. Yeah. So do you think we could turn it around so everyone else can see everything?
Oh,
Here, we'll go and give you a little 360 tour of where we're sitting.
Oh, yeah, sure.
Right now we're at the kitchen table, I guess, and there's the walls of different buttons. There's Otter. Otter can wave. He's one of the habitat technicians. And there's the wall of buttons I was talking about, and the kitchen sink, with all their dishes. Yeah. So.
Where's the bunk room? Is that behind you?
The bunk room is back here. Can you guys see
in there? Really tight quarters. 3 bunks on each side. Back all the way to the ceiling.
How is that going?
It's fine. I sleep really deeply. That's good. I'm probably the worst. I probably snore and talk in my sleep.
Yeah.
Okay, well, thank you guys all so much. You guys want to give a round of applause to our aquanauts here?
Yeah. Oh, and before we go, Amanda and I just wanted to give a shout out to our mom and dad, and to say that my dad's the coolest, and thank you. Yep. All right.
Thank you guys for coming.
Yeah. Thank you guys.
Thank you guys. Bye.
Bye. Yeah. Can we do that?
Elizabeth leaving her phone somewhere.
Close it.
Okay. Cool. Thank you very much for joining us. If you have any questions, you can feel free to come on up and chat with either of us or with Isaac specifically. But otherwise, thank you all.